Greetings IB to the lecturers. My name is Vernon MANGUN and I will be presenting my individual project.
Let us take a look at the overview. The chromatin immunoprecipitation assay, or ChIP assay in short, is a powerful and versatile technique used for probing protein-DNA interactions within the natural chromatin context of the cell. This assay can be used to investigate the interaction between proteins and DNA in the cell and identify multiple proteins associated with a specific region of the genome, or the opposite, to identify many regions of the genome associated with a particular protein. In addition, the ChIP assay can be used to define the spatial and temporal relationship of a particular protein-DNA interaction. Moving on to the molecular basis of ChIP, by its name, we can already take a hint that this tool is used for identifying interaction on DNA. As we can see that there is a chromatin, so we have to know what chromatin is. Chromatin is a substances within a chromosome consisting of DNA and protein, and the major protein in chromatin are histones, which aids in DNA packaging that allow DNA to fit in cell nucleus. This tool uses the concept of immunoprecipitation, which it uses a specific antibody that immobilizes to a solid support such as magnetic particles or agarose beads. Before we go any deeper, we have to know and understand the properties of antibodies and also antigen. An antibody, also known as immunoglobulin, is a large Y-shaped protein produced mainly by plasma cells that is used by the immune system to neutralize pathogens such as pathogenic bacteria and viruses. The antibody recognizes a unique molecule of the pathogen called an antigen. Antigen is a toxin or other foreign substances which induces an immune response in the body, especially the production of antibodies. Antigen is actually a protein. This is the basic steps of ChIP assay. So firstly, the DNA binding proteins are cross-linked to DNA with formaldehyde in vivo. Next, to isolate the chromatin, share DNA along with bound proteins into small fragments. After that, binding antibodies specific to the DNA binding protein to isolate the complex of precipitation, and reverse the cross-linking to release the DNA and digest the protein. After the crosslinks are reversed, the DNA is purified and ready for analysis. Moving on to the next slide, it is about phases in ChIP. So there are 5 phases in ChIP assay altogether. Let's start with the phase 1. It is about lysate preparation and chromatin linkage. So the chromatin linkage is done by formaldehyde. Formaldehyde is the smallest aldehyde, an electrophilic molecule susceptible to chemical attack by a wide range of nucleophilic species of biological interest. So this can aid in the linking of protein and DNA. How it links the DNA? It has step-by-step procedure. So the first step is a nucleophilic group on an amino acid or DNA base forms a covalent bond with formaldehyde, resulting in a methylol adduct, which is then converted to a shift base. The methylol and shift base can decompose rapidly or may be stabilized in a second chemical step involving another functional group, often on another molecule, leading to formation of a methylene bridge. After formaldehyde is added into the sample, glycine will be added next as a formaldehyde quencher. In preparing cell lysate, there are 2 types, which are chemical and physical. Chemical cell lysis uses cell lysis solution. Cell lysis solution are detergent-based buffers and reagent sets that have been optimized for a particular cell lysis application. However, effective cell lysis and protein extraction for different species of organism and different cell and tissue types require different buffer formulations. For example, SDS. And physical cell lysis homogenizer, or also known as blender, is usually used. Moving on to the second phase, which is the DNA shearing by sonication. Sonication is the act of applying sound energy to agitate particles in a sample for various purposes such as extraction of multiple compounds from plants, microalgae, and seaweeds. It can shear DNA into fragments as small as 100 base pairs in length. And the optimal size of chromatin for ChIP assay is usually 150 to 300 base pair. And the machine used is called sonicator. However, there are factors that affecting the results, which are the equipment, the cell type, cell density, cross-linking efficiency, and shearing condition. Moving on to the third phase, which is the immunoprecipitation. This phase is where the concept of immunoprecipitation is used. As we know that the antibody has a specific binding site, means that we need to find the right antibody for the targeted protein. In addition, for the beads used, it's either agarose beads or magnetic beads. It depends on what the researcher prefers, because these two beads has their own advantages and disadvantages. Next phase is reverse cross-linking and DNA purification. The reverse cross-linking can be easily done by heating up the sample up to 95 degrees Celsius and add salt, preferably natrium chloride. This will disrupt the bond between the DNA, formaldehyde, and the protein. For DNA purification, it can be done by following the figure 12. It is usually done by using a DNA fragments kit, and the procedure is usually from the kit itself. The DNA purification procedure is similar to the figure 12. Lastly, DNA amplification and quantification. There are many ways to amplify and quantify, for example, microarray, real-time PCR, and sequencing. Due to many ways to amplify and quantify, there are many types of ChIP assay based on how the DNA is amplified and quantified. For example, ChIP PCR and ChIP sec. For additional knowledge, I added the advantages and disadvantages for chromatin immunoprecipitation assay. The advantages here is that it is efficient precipitation of DNA and protein, and it is very high resolution, up to 175 base pair. And it is very good for non-histone proteins binding weakly or indirectly to DNA. However, the disadvantages are selective nucleus digestion may bias input chromatin, nucleosomes may rearrange during digestion, and it may be inefficient antibody binding due to epitope disruption. Here are the laboratory tools machine involved. For additional knowledge, this is the taxonomy and picture for Arabidopsis thaliana. However, only the leaf part is taken as a sample. In this research, they use formaldehyde and glycerin to cross-link. They also let this mixture sit for 10 minutes in vacuum environment to reduce bubbles that can disrupt the cross-linking process. The picture A is before the cross-linking step. The plant material floats on the surface of the solution and is covered with small air bubbles. The abaxial and adaxial surfaces of the leaves differ in color. And B, after fixation. Platelets should be visibly soaked in solution, slightly transparent and evenly immersed in the cross-linking solution. Next is the optimization of the sonication condition for ChIP analysis. The researcher found, with increasing number of sonification cycles, the peak of chromatin fragment size is shifted towards smaller fragments. So in the experiment shown, the optimal cycle number vary between 20 and 30. They also found that the optimal chromatin fragments ranging from 200 to 600 BP are more abundant in the ChIP sample. For ChIP analysis, the graph A shows the genomic region of FT with black boxes representing exons. 4 fragments spanning regulatory regions of this fluorointegrated gene were tested by designing specific qPCR primers. For the B graph, the EBS binds the FT4 region located close to the transcriptional start site. MYC-EBS corresponds to EBS mutant plants expressing the CMYC tagged version of EBS. MYC denotes transgenic plants expressing the CMYC epitope not fused to any Arabidopsis gene. And for C graph, the EBS mutant plants display higher abundance of H3 histone than wild-type Landsberg erecta in all 4 FT regions assessed. Arrow bars show standard deviation. Chromatin immunoprecipitation is an important technique in the study of protein-gene interaction. With this tool, many things can be discovered and many research can be done in the future to understand the world deeper.